Okay, hello everyone. So we have started with transport in plants. So what we will be seeing, we have already studied diffusion and facilitated diffusion. Now we have already discussed how facilitated diffusion will take place with the help of transport membrane proteins. Facility is given for the molecules which are hydrophilic, which loves water, that can be moved inside with the help of that transport membrane proteins. So some things related to that transport membrane proteins we will be studying now. So again, this is a, you can under this topic only we'll be studying those things. So as I've already told you that plasma membrane is a lipid bilayer. It is a lipid bilayer, and in this lipid bilayer what we have is proteins are embedded, and these proteins, they actually help in movement of molecules which are hydrophilic, because here the membrane is actually lipid bilayer. It is made up of lipid molecules. So all these things already I have discussed. So please watch the other videos and then you can come to this one if you are not understanding like this. But still I am explaining this, right? So these are the proteins. Like this, we have so many proteins which are embedded in the lipid bilayer of the cell membrane. Now, as I told you, facilitated diffusion takes place with this protein. And what is the name of this? We can call it as transport membrane protein, right? Transport membrane protein. Now this protein, it is like it's like gateway, onthara gate iddangidu. It can allow molecules all the time. Okay, if it is a concentration gradient prakara idre, it can allow it. Permission also is required by the plasma membrane. Only molecules which are required by the plant is taken inside. That at least I have told you. So really, about these channels, if you are talking about, then these channels, there are three important terms which you need to remember. We have terms called uniport, okay, and then coport. See here. Now, what is uniport and what is coport? Now, uni means you can understand one. Uni andre single alva. So what happens in uniport? So if there is a movement of a single molecule through this, then this is called uniport. Only single molecule is moving, then we call it as uniport. One other molecule move agta idre adak now uniport anta karitivi. Now by chance there are molecules moving, like two different molecules are moving in the same protein channel, then we call it as coport. Co andre enu together, right? So we call co andre together. So if two molecules are moving together, then we call it as coport. So now uniport andre single molecule movement, coport is some two molecules together, they are moving inside. Now in this coport, again we have two important terminologies to understand. Coport is of two types. In coport we have two types. One is called symport and the other one is antiport. We have coport. In coport we have two types, symport and antiport. Now if you see here, ide example nodi, two molecules are moving from one place to other place together in the same direction. So what you can understand here, two molecules are there, two molecules, and they are moving in the same direction, alla? Both of them are moving in the same direction. So they are moving in the same direction. So this is called symport. Sim andre same, together in the same pathway. So if two molecules are moving in the same pathway, same direction, then we call it as symport. By chance, if the direction changes, like for example, anyone, you can take it like this. If this molecule, the red one what you are seeing is moving from this region to this one, and the blue one, for example, this is moving from here to here, like this. If this is the direction, so one is moving in the one direction, the other one is moving in the opposite direction. If two movement of molecules are taking place in opposite direction, then two molecules, if they are moving in opposite direction, so what do we call it as? Obviously we call it as antiport, opposite direction. So what is this one? Two molecules moving in the same direction. This is symport and molecules moving in the opposite direction, we call it as antipode. And since in both of these things, two molecules are moving, alva, so this comes under copode. And if a single molecule is moving, we call it as unipode. So please remember this terminologies. It's very, very important. What is unipode? What is copode? And out of this copode has types called sympode and antipode. And sympode, antipode, they have been classified based on the direction where molecules are moving, same direction or opposite direction, right? So these are the simple terminologies related to this. Now there is one more thing which we need to discuss here. So remember these channels, there are channels, we call them channels also, no? Transport membrane proteins are also called as channels. Now these channels, it can be open all the time. There are some channels, there are some protein channels which can be open all the time. But there are some channels which are closed. Okay, we have different terminologies for that also. Now sometimes transport membrane proteins are always open, some are always open, and means easily it can allow the movement if they are open. Some are controlled. That means whenever it is required, that time it will be opened. Whenever it is not required, that time it will be closed. So channels are of two types. It can be open, it can be controlled, it can be gated, we say. Okay, so many different types of channels we have. So now here in this, importantly, we have to remember something called as porins. Please remember this one, porins. Now what are porins? So porins are nothing but the same protein molecules which have larger pore size. Andre ಈ protein molecule ಅಲ್ಲಿ ಒಂದು pore ಇರುತ್ತೆ. You can consider it as a hole or something like this. It's just like a movement of the molecules through this till here, like this. Hinge. So this pore size, if it is more, then that protein is called porin. So porins are nothing but proteins which have more pore size. So if size is more of porins, then what will happen? Molecules of bigger size also can move faster into that porins. So porins are basically nothing but transport membrane proteins only, but only thing is their size of that pore is larger. They have more pore size, and you can consider it as large pore size. Pore size jasti idre, then those proteins are called porins. Now here there is very important concept of terminology there, that is called aquaporins. If porin is a protein which is allowing size of bigger sizes, then we have term called aquaporins anta ide. Now what are aquaporins? Porins, you know what are porins? Now aqua means water. So porins which actually allows the movement of water, obviously their size should be more, because it is a lipid bilayer and obviously water hating it is. So for to movement of water should take place andre porins ದೊಡ್ಡದೇ ಇರಬೇಕು and size ಇನ್ನೂ ಜಾಸ್ತಿನೇ ಇರಬೇಕು, and those porins are called aquaporins. Understood? Now even though aquaporins are there, water is moving through aquaporin. Most of the thing what plants require is water only. ಜಾಸ್ತಿ plants water ಬೇಕು, right? So aquaporins, they form a channel totally. Remember this very important term it is. Remember, it is a neat question also. 8 different aquaporins form a water channel. Andre, see, aquaporins actually are proteins which allow water. Such type of 8 aquaporins are there which are different from each other. They are not same, they all are different. So 8 different aquaporins togetherly, they form a channel for water. We call it as water channel. So whenever question comes of what is water channel, so remember water channel is made up of 8 different aquaporins. This is what you have to remember. Okay, so this is regarding this completes facilitated diffusion along with some extra terminologies associated with that. Also unipode, copode, sympode and antipode. Very, very important these terms are. So please concentrate properly, understand it properly. Thank you.